Thank you, Alison, for the really nice introduction. And thank you all for coming to the seminar. I'm really excited to be here with you today. I am the director of the Material Robotics Lab here at BU, and I'm excited to be telling you a little bit about the research that we have going on. Now, we've been at BU for about 5 years. So I want to start with what really motivates me and my students to wake up every morning and get into the lab and do, work and keep ourselves, I guess, motivated, which is solving open societal challenges in healthcare. So addressing open societal challenges when it comes to making robots that can enable easier, more intuitive surgical procedures to perform, and also enable new surgical procedures that are not possible to perform today. Maybe some of you are familiar with the idea of open surgery, where basically a patient is going to be literally cut open and the surgeon has full awareness of what is going on within the surgical environment, so they have direct visualization of the environment, direct haptic feedback, because they can touch the organs and they can feel what is going on. But really what we want to do is move towards more of a minimally invasive surgical scenario where you don't really have to perform cuts to access the surgical target area, but you navigate to that surgical target area in a minimally invasive way, which means that you're accessing the target area through natural orifices, for example. So think about navigating to the brain by accessing your nostrils, or accessing through the mouth and reaching the lungs and going and exploring the lungs to look for a tumor, take a biopsy, that type of thing. So this is great because patients don't have any postoperative complications, scarring, infections, and they basically can walk outside of the operative room the same day. However, there are many other problems associated with this, which basically have to deal with the complexity of performing that procedure. So surgeons have to undergo years of training because steering and controlling these flexible instruments is a little bit more complicated to do. And so we want to build robots that can do these procedures better and help surgeons perform their day-to-day job better, return some level of information that they don't really have, and return some levels of dexterity at the distal end of those instrumentation. So you have to imagine, like, if you want to control a hose and you want to control the flexible tip of that instrument, it's really difficult to do it, when you have such long, flexible instruments like endoscopes, for example. And so these clinical applications, these medical applications, I would say, really give us the opportunity of, these are case studies that are really good for roboticists like engineers, roboticists like myself, because we are really interested in solving fundamental questions and open challenges in robotics associated, for example, with robotic navigation, robotic manipulation. So, for instance, what is more, challenging than navigating into environments like these ones, to the colon, is a very unstructured, flexible environment. When you have a robot navigating through, it stretches and moves. And same thing here. Navigating inside the lungs, it's really difficult because it's a maze of very different, I would say, tubes, branches of bronchi that you have to navigate into to get to the location where the tumor is located, for example. And so navigation is a really challenging problem in these fields, in these application scenarios. Manipulation too. You can think of, you want to access a tumor located in the brain, maybe in a deep-seated location within the brain, and you want to remove that tumor. But how are you going to be doing that, preserving the healthy tissue surrounding the brain? Because at the end of the day, yes, you want to treat the tumor, but you don't want that patient to wake up with some neuromotor functionalities impaired or neurocognitive functionalities, and it's super crucial how you interact and manipulate the brain tissue. And so these are all really great case studies that allow us to tackle fundamental robotics challenges, especially related to how we are going to be building these robots. I'm a really, my lab is really hardware design focused because we cannot really buy a commercial robot and just use that for these types of applications. Our focus on a day-to-day basis is talk to our collaborators, talk to our surgeons that are collaborating with us, understand the clinical problems that they have on a daily basis, and engineer a robotic solution to it. So we have to start from scratch when we do that. And sometimes this also means that we have to develop the manufacturing technologies to build these robots. And so we have to think about how we're going to be building actuators and sensors, how we're going to be manufacturing them and integrating them inside our robots. And we have to think about how these robots are going to transmit forces onto the tissue. We want to have effective robotic systems to do that. And so in my lab, we have been investigating 3 classes of different soft surgical robots to do that, which are soft continuum robots, soft reactive skins, and in this part of the talk, I'm going to be focusing mostly on how we integrate soft sensors inside our robots, specifically focusing on soft optical sensing, and then hybrid soft rigid robotic mechanisms. So robots that basically want to combine the best of both worlds when it comes to soft robotics, which has many advantages. So you have soft materials that can easily and safely interact with the human body, but at the same time, you have rigid components that allow these soft robots to perform a little bit better and sort of like have higher force transmission or stiffening, or even being able to be folded down into really tiny compact configurations and then use origami-inspired mechanisms to unfold, open up, pop up the robot, and then perform a surgical task. So with soft continuum robots, we've been mostly focusing on interventional bronchoscopy. And the reason why that is that lung cancer is the deadliest cancer today. It kills more patients than prostate, breast, and colon cancer combined. And so one of the reasons why that is that it is very difficult to navigate to the peripheral part of the lungs, so over here. And that's where approximately 80% of lung cancers are located. And if you were able to reach that position early on and were able to diagnose cancer early on and treat it early on, of course, you would be able to achieve a higher survival rate. And so we've been working on this robot that was actually published in Soft Robotics a couple of years ago. And I hope you can appreciate how much smaller it is with respect to, for example, a traditional bronchoscope over here. You can even pass it through the working channel of a traditional bronchoscope. So this is actually the smallest, most flexible robotic bronchoscope these days. It has a diameter of 2.4 millimeters. And in this diameter, we were able to integrate 1 degree of freedom bending, actuation mechanism, a micro camera, and 2 working channels. 2 working channels are beneficial because when you are at the surgical target area right here, and you start poking into the tissue and taking biopsies and so on, of course, you'll have blood, you'll have bleeding, and that, of course, gets in front of your camera. So having 2 working channels basically restores that ability of doing multiple tasks. And so as you're poking the tissue, you can probably clean up the camera, aspirate, and do something more useful without having to pass instruments back and forth and switch instruments with only 1 working channel. So I won't go too much, in the interest of time, I won't go too much into the mechanical characterizations of this robot and the workspace, but really what I want to talk about is, what we did in terms of ex-vivo tests, so making sure that we were able, because this robot is soft and is so small, that we were still able to perform tissue puncturing, to perform biopsies, but also we have, this system has 2 working channels so that we could potentially inject drugs into the lungs. And so the reason why we want to do that is that typically when you go screening for lung cancer, you take a biopsy, then they analyze the biopsy, and the patient goes home, and they are waiting a long time to get the results. But then after a few weeks, you hear back, and if it's cancer, you have to go back and do surgery again, right? That's not really effective at all, because at that time, weeks, months will pass, and your lung cancer goes maybe from stage 1 or 2 to stage 3 and 4, and maybe, there is nothing really more to do. Wouldn't it be great if we have robotic technology that would allow us to take a patient in, have them being screened for a biopsy procedure, and then when they wake up, your surgeon would tell you, well, bad news, we found that you have cancer. But the good news is that we have already treated you, and you can come back in 6 months or a year just for follow-up, so that more lives would be saved at the end of the day. And so one thing that we really were excited about with this soft robotic bronchoscope that we created is the fact that also keeps vision sensing integrated at all times during the surgical procedure, which is crucial to help surgeons achieve a better, higher accuracy biopsy and also diagnostic yield. So by using computer vision algorithms, we were able to basically create a robot that is sort of like semi-autonomous. And so the surgeon can basically use a graphic user interface to command the robot to navigate to a specific pre-planned location within the lungs, and they would just click where they want to go. The robot would align into that location, and then it can be pushed and propelled forward. And the other thing, of course, is by using computer vision algorithms, we can help the robot compensate for breathing motion. And so you have to imagine that the lungs are expanding quite a lot as you're breathing, and the surgeon is performing biopsies. And so you have to be sure that you're actually puncturing where the tumor is. And so compensating for that type of motion is very important for us. And so taking it a step forward, what we did was actually we've been creating these soft miniaturized robots, like I said, compensating for breathing motion and taking biopsies. But really, as you're scaling soft robots down, the amount of force that you actually can output through these robots is also scaling down quite substantially. And so we came up with other interesting questions about, for example, well, how are we going to be able to make sure that this robot is capable of puncturing through very effectively? How is it going to be controllable within the lungs as it's performing the procedure? And so over here, this paper that we published very recently this year, this soft robot is capable of deploying soft robotic actuators to anchor itself through the surrounding bronchi anatomy. And that helps the robot be more accurate, but at the same time, it also helps the robot sustain a higher level of force output as we are trying to target, a biopsy. And so over here, basically, I'm going to skip quickly maybe through some of these things, but over here, we were able to demonstrate that we were able to achieve pretty high output forces, which are way above the range of the surgical tasks that we want to do as we are performing biopsy procedures. And we were also able to demonstrate that we have created origami-inspired soft robotic mechanisms that can help the surgeon control the needle deployment inside the lungs to take biopsies, such as, for example, in this case, we have a needle attached to a soft robotic origami-inspired actuator that can basically command the puncturing of the tissue. And next steps over here are going to be integrating also steering, because sometimes when you are at a location when you want to take a biopsy, the tumor is located maybe close by, and so you have to achieve pretty sharp bending angles to make sure that you can puncture your way through. And so in this in vitro test, we have a 3D printed model of a bronchi where we basically have our robot navigating to. At this point, the robot is stabilizing itself around the surrounding anatomy so that it can be more accurate and puncture its way through the lungs. Over here, this is a gelatin model of the lungs. We demonstrated that we are still able to steer the needle in case that's necessary. Of course, we will have also intraoperative imaging, CT scans, as we are performing the procedure, and then we're able to deploy the needle inside the lungs, hopefully to get where the tumor is. And over here, the dye that you're seeing is just a simulated dye that we use to make sure that we're actually puncturing through the lung tissue. So switching gears a little bit, I want to talk a little bit about soft robotic skins, soft reactive skins. And like I mentioned in my introduction, these are soft robots that you can wrap around any type of medical instrumentation to robotize it. So they are dense with soft sensors and actuators. And in this case, these are soft optical sensors my student is pressing upon them, and as you can see, the soft sensor is triggering the inflation of a soft robotic actuator. The way that we are realizing these is just basically we are using different types of soft elastomers, as you can see here. And by playing with the refractive index of these soft elastomers, we're capable of transmitting light through these waveguides that you see over here. And so transmitting light through, and then if you press on these sensors, they will, the sensor response will change, and you're able to track pressure, forces, bending, and a variety of other things that I'm going to talk about. And one application that we had in mind here was interventional colonoscopy. So sometimes in colonoscopy, you know, as you're pushing the colonoscope inside the colon to navigate, you might have excessive force concentration. And of course, if you're a healthy person, it's not too much of a huge deal. But if you're not and you already have some comorbidities, this can potentially cause perforations, bleeding. If you are located in a place in the world where you don't have access to anesthesia or, you know, good large hospitals, you might want to have something like this where you have a soft robot that allows the surgeon to perform a gentler, safer procedure. My dad actually recently underwent a colonoscopy procedure. My family lives in the south of Tuscany in Italy, in a really small town. They don't have anesthesiology in their hospital. So he underwent this colonoscopy procedure, and it was so painful for him, and he's a tough guy, and it was so painful for him that he couldn't withstand the procedure. And so when I told him about the research that we do in my lab, of course he was like, oh, when can I get, you know, your instrument to do this procedure? Unfortunately, not really soon. But anyway, so what we do is basically, like I said, use these sensors to trigger the inflation of these soft robotic actuators, sort of like airbags, if you will, that can help the surgeon distribute these forces and pressures and hopefully have a gentler procedure. I want to show you this in vitro simulated test that we did, where this is a model of a colon where we basically show that if we apply too much force within our colonoscope, the soft robotic actuators are capable of deploying. And we are also capable of, like I said, if we have a very concentrated amount of force, by triggering these soft robotic actuators, we can distribute that around a larger area to make the procedure a little bit more gentle for the patient. And then we took this a step further by amplifying that haptic feedback back to the surgeon. So this is the soft robotic sleeve that we have over here, and as we're pressing on it, we are returning that haptic sensation back to the surgeon as they are wearing a soft robotic glove over here. And we were able to test this together with a surgeon that is collaborating with us at Brigham and Women's Hospital. This person especially is an expert surgeon. We are hoping to start testing on novice people as well. But the goal of this is really providing that level of extra awareness for the clinician such that they can be able to perform better procedures. And then again, for more of a trained person, you would be able to still see an improvement in the procedure. We measured the NASA task load for our surgeon, and we're able to see that the mental and physical workload was diminishing for them as they were performing that procedure. But you can imagine that even if you were taking somebody novice, that would help them, you know, be more trained and be able to perform a better procedure in the long run. So these technologies can not only help people, patients and surgeons as they are performing the procedures, but even can help during training. Regarding haptic feedback, we've been focusing not only on interventional colonoscopy and the project that I just talked about, which is funded by NIH, but also we are tackling other aspects of haptic feedback in remote palpation or robotic-assisted palpation. So by using soft robotics and microfluidics, we are able to manufacture really tiny soft actuators that can be positioned at the fingertips over here, integrated in soft robotic wearable gloves. And what we do is basically, again, by using soft optical sensors. In this case, this is so easy and super cheap to manufacture because these are literally flexible tubes that we are integrating in our sensors, and they are very inexpensive and easy to manufacture. And over here, again, we're showing how we are using microfluidics to create our soft robotic actuators. And so what we do is basically we have this soft sensor that is mounted at the end effector of a robot, and it is basically palpating a tissue model where we have embedded a tumor, so like a higher stiffness material in here. And when we are basically sensing that tumor, these actuators are going to, return that haptic feedback back to the surgeon. And this is a paper actually that we currently have under review. But again, there is so much more that you can do with this soft optical sensing technology that we have developed, and I just want to touch upon quickly two other applications that we're looking into, which are soft optical sensors for shape and contact tracking. So in this case, this is a soft continuum robot with a module over here that has multiple degrees of freedom and a soft robotic gripper mounted at the tip. And we are able to not only track the shape of the robot and the gripper, but also detect if the robot is contacting objects and interacting with the surrounding environment. So how do we do that? This is the module. It has soft optical sensors embedded inside these ones over here. It's only one sensor at the gripper. And by using roughness tuning, we can tune the optical properties of that sensor. So we are roughening the tip of our mold over here, and that roughness actually translates into the soft optical sensor, such that basically we're able to have from only one sensor a multi-modal response. And so the sensor is capable not only to track shape, but also contact. And so as you can see here, when our gripper is moving, we're getting the response that we're supposed to be getting, but then as it's touching an object, we're getting a different type of response, and we can track that contact. And the cool thing about that is that you can track these two different inputs, and how you do that is basically, I want to show you a live plot of this. So as the gripper is deploying and opening, you have this type of response, but then if you have an object placed at the gripper jaws, you basically have a different type of response, which basically gets you that information that you're looking for, whether or not there has been contact, with just one sensor integrated. The cool thing about this robot is we're looking into potentially having it applied in minimal invasive surgical applications. So, for example, a scenario like this in laparoscopy. And so we were able to test this. This is recent work that we currently have under review. We were able to test this in a sort of like pick and place peg transfer task scenario, where we pick up an object and we move it to point A to point B. And recently we've been not only capable of tracking the shape of the gripper and the robot and returning information about whether or not there has been contact, but we are also able to track and sense how much force we are applying onto the object, which is crucial in applications, again, like minimal invasive surgery, but you could think of any other type of application like environmental exploration, any type of really application in robotics where you would benefit from having a really snake-like robot and to manipulate the environment and grasp objects and so on. And then, again, just one really quick other thing that we can do with this technology of soft optical sensing. We have been looking into other types of clinical applications, such as, for example, bleeding detection. This is very important in applications like colonoscopy, for example, where you have a camera located at the distal end of the instrument, but you don't really have any other information about any other sensors along the length of the endoscope. And so you don't have any information about what's going on in the back. And so sometimes, like I said, if you're applying too much force or the patient has any other comorbidities, you might get bleeding. And you end up knowing that there has been bleeding actually only when you're pulling away the instrument, right? And so we've been looking at soft optical sensors that can detect bleeding, and you could think about having them distributed along the length of your tool, of your colonoscope. And so over here we have a soft optical sensor, a U-shaped soft optical sensor, and by using microfluidics, we're capable of basically picking up blood that has, that is in the colon. The blood will enter our microfluidic channel, and the soft optical sensor here will detect that just by measuring the change in refractive index within the microfluidic channel. So the way that this works is, again, you have light going through this waveguide, and when this is analog blood, synthetic blood, and when blood is passing through this microchannel, this sensor will basically pick it up. And the cool thing about this is that we were able to demonstrate that we are capable of measuring different concentrations of blood. And so based on the sensor response and the bandwidth, you can track whether there has been a minor bleeding or there is a hemorrhage going on. And so you can alert surgeons about that. We are able to differentiate between blood and other types of fluids within the body. So for example, in the colon, gastric fluid. And so we know how the sensor is going to respond. And in the future, we're hoping to use this technology as a biosensor for other types of, maybe diagnosis of other morbidities, other illnesses within the colon or in other locations of the body. This is an in vitro test in a simulated colon environment where we were able to validate our sensor working and picking up blood over here. And we're hoping, actually, Aring, my student, is currently doing ex-vivo tests on explanted colon models with our collaborators, and hopefully over the summer and in the fall, we're planning on doing in vivo studies on this, so in live actual porcine models. So very excited about that. And then I want to switch gears one last time and talk about hybrid soft foldable robots. So like I mentioned, over in this area of research that we currently have going on in the Material Robotics Lab, what we are looking at is combining the best of both worlds when it comes to soft robotics for surgical applications and rigid origami-inspired mechanisms. And so these are some of my past works where I was working on basically endoscopic robotic add-ons. So robots that you could put around the tip of an endoscope and they would lie flat as you are navigating your way, for example, in the anatomy, and so not hamper navigation of colonoscopes, for example. But then when you reach the surgical target area, they would unfold, pop up, and help perform some level of surgical task. And so, for example, in this case, expanding and anchoring the colonoscope tip to enable better force application, better force output during procedures, and all sorts of other things. And so this is, what is interesting about this is that, as you probably know, soft materials and rigid materials don't interface really well. And so if you want to build a robot out of this, you need to be very cognizant about how you're going to be bonding these materials together. How are they not going to delaminate? And so we basically developed manufacturing technologies based on soft lithography, laser precision micromachining, and chemical surface modification borrowed from, again, soft lithography realm to create mechanisms that are super tiny. These are my fingers over here. And by using microfluidics, we are able to have these soft, rigid actuators move around. They can do pretty large range of motion. And also we are able of embedding sensing. So achieving proprioceptive actuation. For example, in this case, we have a capacitive sensor. And as our soft micro actuator is expanding, we're capable of tracking the motion of this robotic actuator, but you could potentially also think about for sensing and other types of sensing again. So one of the things that we did with this technology was developing, like I mentioned, robotic add-ons. So for example, multi-degrees of freedom robotic arms that could be placed at the tip of a colonoscope to perform tissue manipulation, and especially one thing that is called tissue retraction. So in this case over here, you can see that we have a vacuum gripper as the end effector of the robot, and the arm is basically helping lifting this tissue up so that we can perform tissue biopsy and so on. So that was part of my previous work as a postdoc at Harvard, and I was approached by a neurosurgeon when I moved to BU who was very excited about this technology and told me, oh, can we do something for, you know, neurosurgical procedures on a daily basis? I'm trying to access tumors located in a very deep position within the brain, and like I mentioned, they can reach that tumor, but then postoperatively they have complications for the patient, right? And the reason why they have these complications is that basically they're using stainless steel spatulas that are pretty rigid, and they have no sensing, of course, embedded onto them. And so really the procedure looks brutal. These instruments are medieval, if you will, right? So the surgeon doesn't really have much information about how much force they are applying onto the tissue. Why is there not a robotic technology that can make these procedures, you know, more safer, I would say? So we have developed this soft origami-inspired robotic retractor that can basically squeeze into a really tiny configuration and be inserted inside the brain by passing through your nostrils or maybe a burr hole in the skull, and then create a working channel for the surgeons to operate where they can insert instruments and they can see the tumor that is located inside. So of course we were able to characterize the system, but what's most important is that we validated it in an in vitro simulation procedure where we were able to see that, of course, the robot, because it's soft and it has multiple, like a larger surface, it can interact with brain tissue in a safer way and exert forces at a level that is more controlled than traditional rigid instrumentation. And so that was something that we were really excited about, and hopefully we're moving forward. And right now actually, we are looking at integrating sensors on this structure too, to not only measure the forces in a continuous way throughout the procedure, but also what we want to look at is integrating sensors that can track, for example, brain activity during the procedure. And this is really exciting because you can have really have a robot that is interacting with the human and the tissue and return information about, for example, physiological entities, physiological information to the surgeon as they are doing the procedure, and hopefully be even more safe when it comes to performing neurosurgery. And last but not least, I just want to mention quickly recent work that we currently are doing on soft rigid robotic systems, and this is work by my PhD student Brian, who has developed a really interesting manufacturing technology, again based on laser precision micromachining and chemical surface modification of soft materials, to make sure that he's able of integrating these soft origami-inspired actuators inside origami rigid structures. And the way he's doing that is by having everything, so there is no manual assembly of these soft actuators. Everything is really sort of like already thought about as a batch manufacturing procedure. So you can scale these down as much as you like. You can easily assemble them pretty quickly without error, without human error. And the cool thing about that is that you can integrate, again, sensing. In this case, we're looking at ionic resistive sensing, and we are using the fluid that we are using for actuation also as a sensing medium. And so, again, combining all the pieces of a puzzle when it comes to robotic actuation, sensing, and controls. And Brian was able to develop different types of joints and degrees of freedom. So over here we have linear, rotation, rototranslational combined, and this is the response of the sensor. And because these are, again, hybrid robots, we can use the benefits of both worlds. And so this goes back into basically having rigid robots, and we all know how to predict the motion of rigid robots and model them, right? And so when we are able of doing that, we can track, predict their position. We have sensors embedded that return us every type of information we need to know about where the robot is in space, how it is moving, and so on. Over here, we're tracking with an EM tracker, with an electromagnetic tracker, the position of the robot, but really the response that you're seeing here is the response of the ionic sensors that we have in our robot, integrated in our robots. Unfortunately, one of the problems here is that, as you can see, there is wiring problems that we still have to figure out. Many wires are coming off of our robot. But one challenge that we were able to solve with this manufacturing technique, hopefully you'll hear more about this because this paper is in preparation, is the integration of fluidic lines. And so you don't see any fluidic lines over here because they are all integrated within the robot. Yeah, so I think I'm on time. I just want to conclude by thanking, well, of course, big shout out to my lab, my students. Over here, I think I mostly talked about the work that Max, who is my most senior graduate student, has been doing, and he will be graduating over the summer. Daniel, Rukaya, Viola, and a bunch of others, Franco, Brian, and Erin, and then a bunch of other master's students and undergraduate students in the lab. So big thanks to them, really lucky to be working with them. And of course, I want to thank my sponsors and collaborators, and thank you for your attention. And I really appreciated you coming to the seminar. Thank you so much for having me.
Great, thanks. Actually, can I kick it off? I was curious in these medical applications about fluidic or air lines going into the body. Are there concerns about bursting, like, the actual drive mechanisms for the robot compared to things that you actually want to deliver? And what kind of challenges are there for safety?
Absolutely, yeah, that's a great question. So the pressures that we are running in our robots are relatively low pressures, but because, these actuators, if they were to pop, yes, you would have like fluids squirting out of the actuator, I guess. So one of the reasons why we're looking at specific applications like, for example, colonoscopy, where we're using saline solution to actuate our robots, is that it would be safe if you had a leak, within the colon. Basically, interventional colonoscopy surgeons are already using those type of pressurized fluidic lines with saline solution. So it's something that has already been used in this field, which we consider, being safe. For interventional bronchoscopy applications, we are using air. You don't want to have like fluids within the lungs, for sure. So using air, again, gives you that level of safety. In terms of pressures, again, and forces that you would be expecting if one actuator were to fail, these are, if you're looking at forces that are applied typically to the lung tissue during rigid bronchoscopy, they are in a very high range. So we're talking tens of newtons, and so that gives us enough room to think that these actuators would be safe to use in this scenario. But then again, definitely there is more work to do, and we're hoping to be validating these systems in vivo, like I mentioned, really soon on live animal models to evaluate safety concerns, and so that's one thing. On the other hand, because one thing that I probably haven't stressed enough is that these robots that I talked about today are all conceived with having in our mind the idea of using them as disposable. So no need to sterilize them and have them undergo many procedures, and that gives us a little bit more room for safety because they will not be used for many cycles. And so hopefully the risk of an actuator failing is lowered by that.
Great, thank you. Other questions? I'll go here and then here.
Thank you for the talk. It was very interesting. I was curious for your bronchoscopy applications. I think a lot of the actuators have 1 degree of freedom. In general, for bronchoscopy, is 1 degree of freedom enough, or is there any benefit if you have additional?
Yeah, so currently in bronchoscopy, so a system like, for example, this one that I'm showing over here, a traditional bronchoscope has 1 degree of freedom. For traditional endoscopes in general, you typically have 1 degree of freedom, unless we're talking about colonoscopes that are slightly larger, where you can get 2 degrees of freedom, pitch and yaw. Typically, you only have 1 degree of freedom with traditional instruments. And so what you end up doing is the clinician rotates from the outside to achieve a full range of motion. With our robot, we obviously want to do better, we want to do more, and so what we are looking at is potentially using, let's see if I have it here. Yeah, so we have multiple working channels that we can integrate into the section of our robot. For this first prototype that we developed, we had a working channel for the camera, 2 working channels for instruments, and 1 working channel over here, which is actually closed up at the end of the robot, and it works as a pneumatic chamber that helps us steer the robot around. But there is no reason why you couldn't have an additional working channel somewhere else to have 2 degrees of freedom. So definitely, yes, we would love to have that. And at this time is more of a, I guess, compromise, because you're looking at miniaturizing these as much as possible to get to remote locations within the lungs. And so it's a compromise of how much complexity you want to have on board of the robot. But hopefully, yeah, that would be possible.
Thank you.
Yeah.
I work at Intuitive, but not specifically on our ion products, so I don't know that much about the lung geometry. I was wondering if you could elaborate. I saw your sort of neck down actuator, and you were talking about how most cancer nodules are sort of deep into the lung. How does the geometry on this actuator that you've created, I guess, compare to those very, very small orifices at the edge of the lung? Sort of, I guess, how deep does it penetrate?
Oh, yeah. So far, and yet we have to test it on live animal models. But so far, basically, if you're looking at a traditional bronchoscope, they typically range in diameter about 6 millimeters or so. There are some smaller ones, sort of like pediatric bronchoscopes. However, they don't really have, so the smaller you go, and you don't really have working channels, and so they're traditionally just for diagnostic procedures, but not really like interventional tasks. You don't have any instrumentation passing through. So as you're scaling down with these instruments, you're losing some level of capability. And so with traditional bronchoscopes, you can reach, these are called generations. So every time you have a bifurcation, you call it generation. And so you would be able to probably reach 2 or 3 generations. And then you have to basically deploy tools without direct vision guidance from the camera, but under X-ray screening and navigation. And so if you were to push further, sometimes you can, but it has been proven that the stress that you're applying onto the tissue is way too high. And so post-procedure patients typically have postoperative complications and tissue trauma and bleeding and scarring. And again, it's not ideal when you have a person that already has lung cancer, right? You don't want to go that way. So with our system, the diameter is less than a half, almost. And so that is a good thing, because we hope that we are going to be able to proceed further than the 3rd generation on a live animal model without causing that much tissue trauma, because the robot is soft, and hopefully because it's automated, because it's has robotic technology that allow you to steer the robot and propel it forward and see what's going on inside. You should be able to see less tissue trauma as well. We're hoping to basically assess these capabilities ex vivo first before we're moving to live animal models, and so look at scratches on the explanted tissue, whether or not there has been trauma before we move forward.
We have time for one more question.
So great talk. I'm very interested into your soft gripper work that you showed, and I wonder what would it take to integrate it, maybe somehow with existing commercial rigid surgical robot systems such as the Da Vinci.
Soft gripper. So are you talking about
the ones that can detect contacts and that was used to deliver haptic feedback to the surgeon? So.
Oh yeah, yep. Let me. Is it this one?
No, actually, it's a gripper, the one that was used for pick and place.
The soft gripper with
Oh.
the blue one.
Oh, okay. Hold on. Is it over here? This one? Or maybe you're referring to. Okay, gotcha.
Yes,
correct. Got it. So the question is, how easy would it be to connect this to like a Da Vinci and the wrist?
Yes.
Yeah. So over here, as you can see, I'm hoping that you can see something over here. There's a little bit of light going through the waveguide, right? Here and here, there are optical sensors. There you go, over here. So these optical sensors, the good thing about them is that you can manufacture them, for example, with soft lithography and micro fabrication techniques. And so you can scale these down. So for a system like this one, I believe the diameter of our sensor was about 100 or 200 microns, but you can go even lower than that. And we have also been able to prove that this technology we can use with micro tubings, such as these ones over here. We're still able to guide the light through. These tubings are quite large, but we've been recently able to demonstrate that, with really tiny tubings, like 100 or 200 microns medical tubing, you can still achieve these capabilities of soft optical sensors. So I think one thing that you need to consider when you're talking about an instrument like the Da Vinci is definitely you want to scale the sensor down as much as possible to make sure that it's going to be able to integrate on the gripper of the system. And it's definitely possible because you don't have, this is an optical sensor, so there's only light passing through the sensor, no current, no voltage, no temperature, and so on. And so, yes, it would be definitely interesting to look at that, and it could be definitely possible, yes.
Okay, with that, I think we're at time. So thank you all very much for your questions, and thank you, Sheila, for the great talk.
Thank you so much for having me.